pathogen, come and destroy me. This is autocrine signaling. It can also do this when it is going to, a cell is going through cell death. It can signal, it can release molecules that are going to signal the cell, the receptors on the cell membrane, to initiate apoptosis, to initiate cell death. The other form of signaling we have there is juxtacrine signaling. The second one is autocrine. We have autocrine. And the third one is juxtacrine. Juxtacrine signaling. We are saying juxtacrine signaling is signaling between two adjacent cells. Two cells that are together. Two cells that are together. We are saying signals are transmitted across cell membrane. In plants, we have this is very common, because remember, plant cells are attached together, having pathways called plasmodesmata within them. So signals can come from one cell and go into the other cell and pass on and elicit a response in the next cell. This is juxtacrine signaling. Please take me to the next slide. Yes, juxtacrine signaling is signaling between adjacent cells. One cell produces a signal, and that signal passes through the cell membrane into the next cell. So we are saying these are transmitted along cell membranes via components integral to the cell membrane. A good example, these are also called contact dependent. Yes, juxtacrine signaling is also called contact dependent. Contact dependent. In other words, for a cell to qualify as a juxtacrine, for a signal to qualify as a juxtacrine signal, it must have contact. The two cells must be in contact with each other. A good example of this is the cardiac muscle, the muscle of the heart, the cardiac muscle. Please take me to the next slide. The cardiac muscle. Yes. Now, when you look at that muscle right there, there are three images that I want to emphasize. When you look at that image right there, we have what we call gap junctions. When you look at the upper right corner, yes, upper right corner, we have those gap junctions right there. We have desmosomes. Yes, we have opposing plasma membranes. Now, adjacent cells are connected by those intercalated discs, but through those gap junctions, stimuli signals can pass through quickly. This is why the heart muscle is able to respond as fast as it can, because it uses juxtacrine signaling. Signals pass through cell membranes directly into the next cells. They pass from one cell to the next cell straight away through the membrane of one cell. That is why the cardiac muscle is able to respond. If you looked at how the heart muscle is excited, when we are looking at the cardiac cycle, we looked at how the stimulation of the heart is stimulated from the sun to the AVN. All of this is able to happen because the cardiac muscles utilize juxtacrine signaling. So a signal will come from one cell, go through the gap junctions, and go to the next cell as fast as possible. So one of the advantages of juxtacrine signaling is that it is very, very fast. Very, very fast. So that's what I wanted to emphasize there. An example of that is that when you look at that signal right there can travel very fast, implying that if one cell is excited on the cardiac muscle, most of these cells are going to become excited at almost the same time. Another type of chemical signaling we have is paracrine. Is paracrine signaling. Paracrine signaling. Paracrine signaling is signaling of a cell within the vicinity of another cell. If this is the cell right here, it produces a chemical that floats and then comes to the target cells here. Paracrine signaling. A cell produces a signal to another cell within the same vicinity. Yes, within the same vicinity. So we are saying paracrine signals target cells in the vicinity of the secreting cell. This cell that is secreting secretes to cells that are near it. Yes, that are near it. but it does not go through the cell membrane like juxtacrine. Do you remember the juxtacrine signaling? Signals go through the cell membrane directly into the next cell. Autocrine signaling, a cell releases its signals, and these signals go back, they move out, they are secreted out, and then come back to the cell itself. Yes? Intracrine signaling, cells produce signals, but those signals remain within the cell. However, paracrine signaling is now where we look at a cell produces a chemical that the two are not intouched, but they are close to each other. The two are not intact, but they are close to each other. So in this case, this signal travels for a short distance until it attaches to the cell within the closest vicinity. That is paracrine signaling. The extreme of this is endocrine signaling. Endocrine signaling. Yes, so please take me back to that slide. No, the next slide. Paracrine signals target cells in the vicinity of the emitting cell. They target cell in the vicinity of the emitting cell. We have one cell close to the next one producing a signal that is going to attach to the signal to the cell that is directly adjacent to it. Endocrine signals on the other side, on the other hand, target distant cells. Yes? Endocrine signals target distant cells. We know of endocrine organs. We are going to be looking at the endocrine system tomorrow. We are saying endocrine signals, for them, a signal is going to be produced in the brain, for example, and it's going to have effect on the ovaries. It's going to have effect on the testes. That is long distance. It is going to, so endocrine signaling, signals are produced by the secretory gland, and they are poured into blood. They are poured into blood, transported by blood to where the target cells are going to be. So endocrine signaling involves long distance signaling. We are saying there, endocrine cells produce hormones that travel through blood to reach all parts of the body. It is therefore very important that a cell should be able to identify which signals are its own and which signals are not its own. This is why cells require to have specific target cells. Yes? Okay, so let us look at that next slide. We have some illustrations. That next slide has some illustrations. Yes, The first image you see there is autocrine signaling. Yes? The first image you see there is autocrine signaling. Autocrine. A cell produces a signal, it is secreted, and then it has effect on the exact. It has effect on the exact cell it has come from. That is autocrine, where a cell will target itself. The next one is juxtacrine signaling. You can see juxtacrine signaling. A cell targets a cell connected to it by gap junction. The cell is connected to the target cell, and the signals simply go through the junctions between the two into the target cell. The third one is paracrine signaling. You can see that the two cells are close to each other. Yes, paracrine signaling. A cell will target a nearby cell. A cell will target a nearby cell. And finally, we have endocrine signaling. Endocrine signaling, the signaling cell produces signals, ligands that travel through blood to the target cell. They travel through blood to the target cell. We can identify these three things. So we can identify signaling. We can classify signaling. 3 steps. We have direct contact. We have direct contact where we have juxtacrine signaling. We have signaling over short distances where we have paracrine signaling. And then we have over large distances. We can classify signaling depending on distance. Over short distances and over long distances. So if you ever forget, simply remember there are three classifications. If we have direct contact, we have over short distances, and we have over long distances. We have juxtacrine, where we said signals are going to go through gap junctions. Yes. And then we have over short distances, where we talked about the paracrine signaling. Yes, paracrine signaling, where we have molecules a cell, a stimulating cell being near the effector cell. Then we have over large distances where we are going to have a signal traveling through blood to go to where it is going to have effect from. So after we have understood this, after we have understood how signaling happens, we can therefore appreciate the different types of signaling molecules. We can appreciate the different types of the signaling molecules. Now, we have three major types. We have three important classes. The first one is hormones. We have hormones. Yes, we have hormones. We have neurotransmitters. Excuse me. Neurotransmitters and cytokines. We have hormones, neurotransmitters, and cytokines. Yes? Okay, hormones. These are the major signaling molecules. Yes? These are the major. Please take me to the next slide. Take me to the next slide. Hormones are the major. They are the most abundant. They are the key part of chemical signaling. Most organs, most cells use hormones. Most cells use hormones. They are the major signaling molecules of the endocrine system. Yes? Neurotransmitters are commonly found in the nervous system. We shall be looking at them a bit later. We have cytokines. These ones are found commonly in the immune system. You have looked at those ones when you are looking at transport, blood. We looked at immunity. So you probably have had an idea of that. They majorly operate as paracrine or juxtacrine. So hormones are what we are going to focus on today. We are going to focus on hormones today. So signaling molecules can fall in different groups. They can fall in different groups. Particularly hormones. They can be lipids, they can be phospholipids, they can be proteins, and they can be ions. Yes, we can have calcium ions. They can be lipids, phospholipids, proteins, they can be ions. And all of these things determine where a signaling molecule will be. We know, for example, that cell membrane is made up of a phospholipid bilayer. And this bilayer does not allow hydrophilic substances to go through it. Very important. Therefore, if a signaling molecule is hydrophilic, it is not going to be able to go through the cell membrane. However, molecules that are small and hydrophobic find it easy to go through the cell membrane and are therefore intracellular signals. So we are going to see, because of the difference in structures, different signaling molecules have effect on different places. If we go to the next slide right there. If we go to the next slide, we are saying signaling molecules can belong to several classes. We have lipids, we have phospholipids, amino acids, we have proteins, we have gases. Yes, we have carbon dioxide. It can be a signaling molecule. We have different classes. We have phospholipids, amino acids, monoamines, proteins, glycoproteins, lipids. Write some of these things down. Yes, they can help you to remind yourself. Now, for a molecule to be a surface receptor, it has the following qualities. It must be large and hydrophilic. Yes, large and hydrophilic molecules are unable to go through the cell membrane, and therefore must take use of the receptors that are on the cell membrane to have effect. Examples we have there: vasopressin, yes, the thyroid hormone. And then we have acetylcholine. We are going to look at some of this in detail. Molecules that are hydrophilic and large do not go through the cell surface membrane. However, those that are small and hydrophobic, go through the cell membrane. Because remember, the cell surface membrane is a phospholipid bilayer. Molecules that go through need to be, one, hydrophobic, and they need to be small so that they can easily diffuse through. We have calciferol, we have thyroid hormones, and the glucocorticoids. These ones go through and have their response intracellular. Now, looking at the signaling molecules is one thing, but we also need to look at the different types of receptors that we have. The molecules, like I said, attach on the receptors. So if we look at the signaling molecules, we need to pay attention, the same level of attention to the signaling receptors. So let us look at the receptors. Let's look at the receptors. We have two types of receptors. We have cell surface receptors and intracellular receptors. Yes? These ones receive information. These receive information. Some of them are found, when you look at that illustration, we have some that are intracellular. They are found inside the cell. They are found inside the cell after the plasma membrane. And for an intracellular receptor, the signaling molecule needs to be able to go through the cell membrane. For an intracellular receptor, the signaling molecule must be able to go through the cell membrane. A good example of this molecule is estrogen. Estrogen has the ability, because it is small and hydrophobic. Note those words. If you are going to go inside the cell as a signaling molecule, you have to be small and hydrophobic. Large and hydrophilic molecules are unable to elicit response from within the cell. So small molecules like estrogen go through the plasma membrane into the cytoplasm, attach on an intracellular receptor. This will in turn go into the nucleus and elicit transcription, and we shall have protein synthesis starting from there. However, most of them are not intracellular. Yes, most of them are not intracellular. So when we look at that right there, that is an illustration of how intracellular receptors work. A chemical will come in, diffuse through, attach on the intracellular receptor, and then the intracellular receptor will be activated to enter into the nucleus and have effect on the DNA. Let's go to the next slide and look at extracellular receptors with cell surface receptors. Cell surface receptors are the most common. Cell surface receptors are the most common. They rely on two major mechanisms. They rely on phosphorylation cascades and second messenger pathways. These ones are unable, because these cells are unable to go through the cell membrane. So we are saying the target receptor is expressed on the membrane. Yes, and I said earlier that when this cell attaches onto the receptor, it causes a change in the conformation of the receptor. When the signaling molecule attaches onto the receptor, it causes a conformation, a change in the shape of the intracellular receptor, resulting into a response. Yes, and in most cases, there are two types of responses. We can have a second messenger, or we can have a phosphorylation cascade happening, where we have ATP being added on different proteins, and these proteins also cause phosphorylation of other proteins, and these molecules also cause phosphorylation of other molecules until we arrive at the second receptor. So what you see there is an example of a cell surface receptor. We have a ligand coming. When it attaches to it, the ligand will change shape. Yes, will change conformation and result into an intracellular response. Yes, we have the ligand. It doesn't enter into the cell, the cell surface receptor, and then results into an intracellular response. Some of these responses, a good example is the second messenger pathway. Where we have cyclic AMP. Okay, so after we have looked at the receptors and the signals, we can now look at signal transduction. We can look at how signals are converted from being extracellular to being intracellular signals. When you receive light, how is your body able to transport that light to become an information that will be interpreted by your eyes? So we are saying signal transduction is a process by which a chemical or physical signal is transmitted to a cell, yes, resulting into a cellular response. Yeah, there are some things I've skipped that I'm going to emphasize. Yes, a cell receives the process by which a signal, yes, can be chemical or physical, will be transmitted through the cell by a series of different events that is going to result into a cellular response. A signal received by a cell changed by different signals, by different into a cellular response. We have two types. We can have second messenger pathways or a phosphorylation cascade. Where in a phosphorylation cascade is where we are going to have catalyzing of different proteins to form phosphorylized proteins, and these ones will also continue catalyzing others, like that. Okay. Now, in this case, we have terms that we can use to help us understand. That statement right there tells us some of them. The first one is the ligands are going to be messengers, the first messengers. These ones carry the information from without and attach themselves on the cell surface receptor and cause the first effector to change conformation. Yes? When they cause they cause the transducer, which is the receptor, to change conformation. The transducer then passes on the information to the second messenger. Yes? The second messenger then transports the information to the primary effector. The second messenger then transports information to the primary effector. We are going to see this a bit later. And then the primary effector takes it through a pathway that leads it to the secondary effector until we have a response. So if you take me to the next slide, I would like to explain what I've just said right there. Take me to the next slide, please. If you take me to that next slide, where we are going to. So when you look at it from this side, from my right hand side, we have the first messenger, which is the hormone, approaching the receptor. Yes, when it approaches the receptor, it activates a protein, GDP. GDP is activated to become GTP. Yes, GDP adenylyl cyclase. Adenylyl cyclase then catalyzes the conversion of ATP into cyclic AMP, which becomes the second messenger. We had the first messenger as the hormone. Yes, we had the first messenger as the hormone. Let me probably try and illustrate that here. Okay, this was attached to this. We have here another effector. Converted to CAMP. Yes, so this is the first effector, the primary effector. This is the ligand, which is the first messenger. So the ligand can be any hormone. The ligand can be any hormone. This is GDP in its inactive form. When the ligand attaches here, yes, it changes the conformation of the receptor, releasing GDP, so that we have GTP, yes, guanoside triphosphate, replacing this diphosphate here. So we have GTP activated to form GDP replaced by GTP, so that we have this relay protein. This one conveys the information now. This protein having GTP conveys information to the effector, adenyl cyclase. This effector then catalyzes the conversion of ATP to cyclic AMP, which is known as the second messenger. The second messenger. Then the second messenger, this cyclic AMP, is going to activate, is going to phosphorylate different protein kinases to form active protein kinases. So we have here cyclic AMP activating inactive protein kinases. Yes, and these kinases here, when they become active, they set off a series of reactions that are going to give us the response of the target cell. This is what we are talking about when we say signal transduction. An information that was first and foremost extracellular. When it attached here, it led to a series of steps that result into a cellular response at the end of the day. We have a hormone, one molecule of the hormone attaching onto this substance here, resulting into the activation of this relay protein, which activates adenylyl cyclase, which then catalyzes the conversion of ATP to cyclic AMP, which is a second messenger. And this cyclic AMP in turn activates inactive protein kinases, to have the effect that we are going to have. So that is what we are talking about. Yes, that is what we are talking about. So all of these things here, this effect can be done by another hormone. We can have another hormone this side, having the same effect but reversing the effects of the first hormone. That is how an organism cells are able to control this effect here. Please take me back to that slide. Yes, so we are saying that hormone elicits a response on GDP. It releases that protein to convert GDP to GTP. GTP, that molecule that has GTP now, activates the enzyme adenylyl cyclase. Adenylyl cyclase converts ATP to cyclic AMP, which is a second messenger. Cyclic AMP phosphorylates inactive kinases to become active protein kinases, and this process continues until we have a response. What is cyclic AMP? Maybe I had not emphasized. What is cyclic AMP? Cyclic AMP, yes, is derived from adenosine triphosphate. Yes, we have ATP, which is adenosine triphosphate, and being converted into cyclic AMP. So you can see that AMP is a derivative of ATP. This one is adenosine monophosphate. Yes? So cyclic AMP is a derivative of adenosine triphosphate. This one is the source of energy for us. But this one here is the most common important second messenger. Yes, it is the most common second messenger. And how does it work? It works by activating different inactive protein kinases, making them to become active. I have its structure on the next slide. We can look at that structure quickly. I have its structure on the next slide. Yes, that is the structure of cyclic AMP. Yes, it's a bit complicated. You may not need to know that. You can look it up from internet. But what you need to know is that it looks exactly like AMP, just that it has that phosphate group attached again to the adenine group, yes, making it a bit cyclic. So we have looked at the second messenger pathways, but I would like us also to look at the phosphorylation cascades. Yes, I want us to look at the phosphorylation cascades. Please take me to the next slide. What is the phosphorylation cascade? It is a sequence of events where one molecule phosphorylates another molecule. We know phosphorylation is adding a phosphate group. So when in this case, instead of having one molecule activating an enzyme, in this case we have a molecule resulting into a series of different stages of phosphorylation. So instead of having the second messenger pathways, some cells can result into having a phosphorylation cascade. Yes? So please let us look at that illustration right there. So we have the signal molecule. The signal molecule. When the signal molecule is, when the ligand attaches on the signal molecule. Yes, when the ligand attaches on the signal molecule right there, we have the signal molecule releasing a relay protein. That protein right there results into the phosphorylation of an inactive protein kinase to become an active protein kinase. The active protein kinase is then also going to activate another inactive protein kinase and phosphorylate it to become an active protein kinase. Are you seeing that? We had the moment the signal touched onto the receptor, a relay molecule was released. This relay molecule that has been released activates, phosphorylates an inactive protein kinase, making it active. Now, this activated protein kinase is also going to phosphorylate another protein kinase. And that phosphorylated protein kinase will also phosphorylate another protein kinase. And that series, the steps will go on until we shall arrive at the secondary effector, until we shall have an active protein that is going to give us the response. What is all this gibberish that I'm talking about? What do we need to know? Yes? The first thing is, cells do not release as much hormone as the response they seem to have. That is why we need these pathways. We need the second messenger pathways. The first reason is because of amplification. If you go back and look at the second messenger pathway, please take me back to that illustration of the second messenger pathway. Yes? That illustration of the second messenger pathway. One second messenger, cyclic AMP, can activate very many inactive protein kinases. That implies that the effect of one hormone on a cell is going to be multiplied in magnitude, such that even when very few molecules of the hormone are released into blood, we are going to have a magnified response in the cells. A good example of this is epinephrine. Yes? A good example of this is epinephrine. Epinephrine has this effect that when you attach it there, you are going to have a magnitude, a great magnitude of glucose molecules produced. Yes, we have a good example of this when we are looking at glucagon. When we are looking at glucagon, it attaches onto that messenger, onto that hormone, activating a series of response that is going to result into glycogen being converted back to glucose. What is the summary of the things we have looked at? If we look at that last slide right there. What is the summary of things we have looked at? We have looked at how cells communicate through different systems. We have looked at the ligands. We have said the ligands can be large and hydrophobic. So it's small and hydrophobic, meaning they can't go through the cell. And they can be large and hydrophilic, meaning they cannot go through. Therefore, they depend entirely on the cell surface receptors. We have looked at the 5 major pathways. We have looked at autocrine, intracrine, juxtacrine, and endocrine. And then we have looked at the different, what those terms actually mean. We have talked about intracrine being in self-stimulation, autocrine being a cell producing a signal outside, yes, and then that signal comes back and activates, has effect on that particular cell. Juxtacrine is contact-dependent signaling. Cells that are intact together produce this signaling together. We had paracrine. A cell that is near, in the close vicinity of one cell, produces a signal that is going to be activated. And then we have endocrine signaling. We have looked at the 2 pathways, second messenger pathway and phosphorylation cascade. Thank you for listening. I have been your teacher, Mr. Wanyaga Solomon. Have a great day.